after isolation of plasmid, now we are running the Agarose gel, 1% Agarose gel, to check the integrity of isolated plasmid DNA by running the 1% Agarose gel. For that, I am making 1% Agarose gel in 1x DI, trace acetic acid and EDTA. This is Agarose, I am going to add it. So I have melted 0.5 gram Agarose in 40 ml 1% 1x DI. Now I am going to add 1 microliter of Ethidium. This whole process of gel making and gel running etc. will be done by only by wearing gloves, because it contains TBR and TBR is highly carcinogenic. In casting assembly, I have casting assembly. I have prepared. Now I am pouring the melted Agarose. I will left it for solidifying. It will get solidified in 5 to 10 minutes. So now gel is solidified, and I am transferring the gel to running tank. And this tank is also contain 1x DI running buffer. Carefully remove the comb. These are the two DNA which we have isolated, one with the manual tape and one with kit. To run these two, this is 5x DNA loading dye. I'm taking 2 microliter of dye for each DNA. Is the DNA isolated from kit. I'm loading 5 microliter of DNA in one well, mixing it properly with the dye. Manual prep DNA. I'm taking 5 microliter from this also, and mixing properly with the loading dye. And then loading the next well. DNA ladder. I'm taking 3 microliter of this. I'm loading the first well. After that,